Hi, everyone. Thank you so much for joining me today. I will be talking about sensory physiology. My name is Lily Solomon, and the date is May 18th, 2024. I will be talking today about chemoreception. This is the sense of both smell and taste. It's the oldest sense from the evolutionary perspective, and it was found that unicellular bacteria uses chemoreception to sense their environment to locate food. Chemoreceptors detect change in chemical compositions of the external environment and turn this into action potential that can be interpreted by the brain. So, the first thing that we're going to be talking about is olfactory, and this is the sense of smell. So, olfactory bulbs are the extension of the forebrain that receive input from the primary olfactory neurons. You can see the olfactory bulb in the upper left corner with the strings hanging down from it. And this is located on the underside of the frontal lobe. The olfactory epithelium lines the nasal cavity with primary sensory neurons, which are also called olfactory sensory neurons. The olfactory nerve synapses with the secondary sensory neuron in the olfactory bulb through the olfactory tract to the olfactory cortex. This ascending pathway from the olfactory bulb leads to the amygdala and the hippocampus, which are part of the limbic system. Something really cool that I wasn't aware of before is that the amygdala and the hippocampus are part of your emotions and memories. And so sometimes when you smell or taste something, it can lead you back to a memory of nostalgia. And so for me, sometimes when I have corn on the cob, it reminds me of my years when I went to camp because there used to be a corn man. And so sometimes when I have corn, I never knew that reason was because it's a part of my amygdala and my hippocampus, and it can trigger that, which I thought was extremely interesting. The brain uses this information from the hundreds of olfactory sensory neurons in different combinations to create a perception of many different smells. And so you're not just looking at one specific neuron that is giving you all this information about your olfactory tract, but in fact, you're looking at many different action potentials and different senses that are allowing you to come up with that specific smell. I thought this was extremely interesting. Next, we'll look at the olfactory signal transduction, and this is how your body receives this information. You start with the surface of the olfactory epithelium. This has knobby terminals that have dendrites and non-motile cilia. As we've recalled from our past lectures, cilia allows for surface transport along the epithelium. And cilia are embedded in the layer of mucus produced by the olfactory glands. These molecules dissolve in and penetrate mucus before they bind to the olfactory receptor proteins on the cilia. And these olfactory receptors are also called G proteins, linked membrane receptors. And these special um, G proteins turned into the intercellular CAMP. I like to call this CAMP. So when you increase CAMP concentration, it opens CAMP-gated cation channels, which depolarizes the cell. And this triggers an action potential that goes to the olfactory bulb to process that smell. So now we will move on to gustation. This is also known as taste. And there are 5 different taste sensations. There's sweet, sour, salty, bitter, and umami. Many people don't know what umami is, but I thought it was a fun fact that in Japanese, it's called deliciousness. So if you've ever heard of MSG, this is a food enhancer, and this is a taste of umami. And it enhances the flavor of food, and they utilize this in many countries. Each of these 5 taste sensations are associated with a physiological process that are associated with nutritious foods. I'm so sorry. Some are associated with nutritious foods like sweet and umami, while others are warning you for toxic components like bitter taste. So, with the kale demonstration I have over here, that has a bitter taste. But if you have something that might be stale or old, it could also have that bitter taste. So, that's just a little bit of warning for you to look out for. So, to be a little bit more specific, sweet, bitter, and umami tastes are part of 2 type 2 taste receptor cells. These cells express multiple G protein coupled receptors, and this also activates gu- gusteucin. I'm sorry if I pronounced that wrong. And this activates multiple signal transduction pathways that release calcium and initiate ATP release. Now, with sour taste, like a lemon, this is a type 3 presynaptic cell responder. And this increases hydrogen and it also changes the pH. So, if you know as acidic taste versus basics, changes it to a more acidic taste. And this releases serotonin in the primary sensory neurons. Finally, we have the salty taste, like the salt that you taste on your pretzel. And these cells are actually not really known for which cells are actually are involved with salt, but what we do know is sodium enters the taste receptors, which depolarize the cell and creates primary sensory neuron firing action potential. So, if we've learned from our past lectures, as you are depolarizing, meaning that sodium is coming in, it hits to the action potential, and this fires sensory to your brain so that you can know what sense that you have. Next, we have taste pathways. These are receptors for taste are also located on taste buds. Taste buds are components of taste receptor cells that are scattered to other regions of the oral cavity as well. I thought it was just on your tongue, but I thought it was extremely interesting that it's throughout your oral cavity. And for some substances to taste, they must be dissolved in saliva and mucus of the mouth. This is kind of similar to the cilia that I was talking about for the olfactory. Interacting taste ligands with membrane proteins create signal transduction cascades to create chemical messenger molecules to release from the TRC. These chemical signals are released from taste receptor cells and activate primary sensory neurons that end at the medulla. This is in your brain. Sensory processing of sensory information, central processing of sensory information compares to the input of taste receptor cells and sensations to create a behavioral response. So basically, if let's say you taste a lemon, this sends its signal to your brain and it creates a behavioral response. So if you don't like lemon, you might take that out of your mouth really quickly, or you might make that sour taste facial expression. And so this is that behavioral response that I was talking about. So now I just want to move on to a fun little study question quiz. I have the answers on here, but if you want to test it yourself, you can cover up the answers and see if that you can get it yourself. So the first one is compare the current models of signal transduction in taste buds for salty, sour ligands and sweet, bitter, umami ligands. This is the hardest question out of the three. So the answer is bitter, sweet, and umami are membrane receptors on type 2 receptor cells with different G proteins receptors and signal transduction pathways for each ligand. Pathways ends with the release of ATP, and these are apparent in enter type 1 support cells. They also enter type 3 presynaptic through channels, and pathways end with a serotonin release. Next, what are the 5 basic tastes and what is the adapted significance of each taste? So the 5 basic tastes we know are sweet, sour, salty, bitter, and umami. And the sweet and umami indicate a nutritious food. The bitter may contain toxins, like I said before. The salty and sour are relative to body osmolarity and pH, like I said as well earlier. Finally, trace the neural pathways involved in olfaction. So it goes from the olfactory receptors to these olfactory bulbs, to the secondary sensory neurons, to high order neurons, olfactory cortex, and then with parallel pathways to the amygdala and hippocampus with those memories to identify what it is. I hope you all enjoyed this and learned a little bit more about chemoreceptors and their importance in the nervous system. I know I did. Thank you so much, and I hope you enjoyed. Thank you.